my academic youth, I attended the North American Paleontology Conference in Washington, D.C. There, the assembled paleontologists were informed of molecular clock evidence that the radiation of metazoans occurred more than 1 billion years ago. I remember that scene and the skepticism in the crowd. 1 billion years? Where was the fossil record? After all, at that point, our oldest metazoan fossil record was that of the Ediacaran biota at around 575 million years ago. Nah, the metazoans could not be that old, and the molecular clock must be keeping bad time. Turns out we were wrong. Molecular phylogenies generally suggest divergence times between the protostomes, things like arthropods, annelids, and flatworms, and the deuterostomes, that include vertebrates and echinoderms, somewhere between 670 million years to maybe as much as 1,500 million years ago. How does this work? The use of molecular clocks is based on an assumption of a constant rate of gene mutation. To make this work, we need to know two things. First, we need the number of genetic mutations that separate different groups of animals or plants, for which we are trying to estimate the timing of evolution. Second, we need to know when at least some of the evolutionary divergences occurred. For this, we use the timing of appearances of specific taxa or morphological features in the fossil record. The fossil traits provide evidence of when particular genes evolved. For instance, the appearance of the gene that controls the formation of simple eyes could be indicated by our first fossil record of eyes and trilobites. The combination of the number of mutations and the amount of time that has passed over which those mutations occurred gives us a rate of mutations. The big assumption in molecular clocks is that the rates are constant and that the fossil dates are accurate and represent maximum ages. Now here's an example. The branching diagram shows the relationships between a number of different groups of organisms represented by clades or families. We have various dates, the stars, and measurements of the number of mutations that have occurred in the branches of the phylogenetic tree. From these data, we estimate that the rate of mutation in the black star group is about 1 mutation in 1 million years. So, given that there are 100 mutations between the common ancestor of both the black and white star groups and the oldest fossil date, that black star at 100 million years, then the common ancestor must have lived about 200 million years ago. In contrast, in the white star group, there are 25 mutations between the common ancestor and the oldest fossil date at 150 million years. So here the mutation rate is a mutation every 2 million years, indicating that the common ancestor lived about 200 million years ago. We're comforted in this example that the different parts of the phylogenetic tree provide the same estimate for the time of origin of the common ancestor. Our assumption of a constant rate of mutation appears to be supported. Now, this is a made-up example, of course. A key point is that the rates of molecular evolution, that is, mutations per million years, do not have to be the same to be useful in all parts of the tree. Instead, the important thing is that the rates of genetic evolution are known pretty well. The results strongly depend upon 4 things. First, we need lots of fossil dates. These dates are needed to give us confidence in the estimate of the mutation rate. Second, we need to know how closely a gene matches something we can actually see in the fossil record. So if we have genetic evidence of when feathers appear, dates on a geologically young group like fossil chickens won't cut it. Instead, we need dates on the first appearance of feathered dinosaurs, since they get feathers before modern birds do. Third, we need dates that are close to the event we want to date. If we are trying to date the appearance of mitochondria, which probably happened 3 billion years ago, then dates closer to the 3 billion year mark are needed. A date, for example, in the first dinosaur a few 100 million years ago will not be very useful. Fourth, we need dates on lots of gene systems. Ideally, we have rates of genetic mutation in mitochondrial genes and nuclear genes and so forth. Different parts of the genome can have very different rates of gene mutation. So the best information will come from having lots of different parts of an organism's genetics. Now, a common problem is that the molecular clock dates are often older than the fossil record would suggest. This mismatch is expected if the molecular dates are dating divergence times in gene systems, which they are supposed to do, rather than the divergence times of fully formed animals. For instance, the gene for eyes, called PAX6, not only codes for really complex eyes, but also for simple patches of light-sensitive tissue, that have, unfortunately, a bad fossil record. Hence, we know that the fossil record for the first complex eye almost certainly is much younger than the time when the PAX6 gene evolved. With these considerations, most fossil molecular clocks find that the metazoan fungal split occurred about 1 billion years ago. In turn, the Cnidarian sponge split happened about 700 million years. A couple of other events are the deuterostome-proterostome split at about 650 million years and the arthropod-mollusk split at about 600 million years. As expected, the molecular ages are almost all older than the fossil record of these groups. Note also that the oldest ages are before the Sturdian glaciation, and most of them are older than the Marinoan glaciation. Hence, metazoans had to pass through at least 2 snowball Earth events to lead up to the present day. Interestingly, the molecular data on the first sponges at 700 million years accords pretty well with the biomarker evidence for sponges at 710 million years. The sponges are one case where there's a pretty close agreement between the fossils and the genes. But for most metazoans, the agreement is terrible with the molecular clocks, saying that metazoans evolved at least 450 million years before we have any kind of metazoan fossil record. And these nagging molecular dates keep coming up in study after study, suggesting that we paleontologists have to go back to the fossil record and look again. So far, we have not found the metazoan, that is the primordial animal named for the first city of in Mesopotamia. Finding these early metazoan fossils should be a high priority on the list of tasks for the next generation of paleontologists. So next we ask, just what were these first metazoans like?